because I can't see you, just let me know. Okay, speak up. Okay, so shall I begin?
Yes, ma'am.
All right. So I'm going to speak today about a paper of mine that was recently published called Multimodal Duetting in a Paleotropical. Can all of you hear me? Yes?
It was a bit patchy at the end. I'm sorry.
The voice, huh? Can all of you hear me? I'm just going to continue, and if anyone can't hear me or if there are any issues, then, and I'll try and speak a little, slower and louder as well. At some point over the next year, I might also apply for a job at NISER, in which case I'll actually come there and give, present this work as part of, my other work in person. So then hopefully I'll get to see you all then, and you'll be able to see sort of a high-res, both in version of this talk and of my. So I'm going to go into the slideshow mode now, right? So that means I'm not going to be able to see any of you. So just speak up if I should go slower or anything. Okay. So this is a bush cricket. It's what's called a pseudo-feline bush cricket, which means it's false leaf. So its wings look exactly like the jackfruit leaves that this animal tends to be found on in the wild. And this animal faces a number of ecological problems. And what I'm interested in is how these insects solve these ecological problems that abound around it. One of the problems that it faces is something called the cocktail party problem. So the cocktail party problem is the problem that all of you will have faced when you're in a crowded room or when you're in some kind of a crowded bus where several people are talking all at the same time, and you can't quite, hear or keep track of one speaker compared to the rest. You have several speakers in front of you. You know that when the sound of the other people gets loud enough, you aren't able to keep track of one speaker. Now, you might find it hard to believe that these bush crickets generate that kind of noise, the noise of a loud bus. But if you go to a tropical rainforest, trust me, you will hear such a din, such a roar of cricket noises. Because there are 20 species of bush cricket and cricket found in this patch of the remote forest in the Western Ghats, which is where I do my field work. And there are some hundreds and hundreds of each representatives, so to speak, of each species of cricket and bush cricket, all calling simultaneously. And some of them can be very, very loud. They can sort of reach the maximum level of loudness that your ears are capable of hearing. So this bush cricket comes from a family of very high frequency broadband callers, and the female auditory systems are also likewise adapted to hear those broadband high frequency calls. But in order to circumvent the cocktail party problem, where there are so many callers of so many different types and tones, this animal seems to have evolved a very unusual solution. That solution is that they produce a very narrow band that is almost a pure tone, low frequency call at 3.2 kilohertz. And this note is, if you like, I've played for you because that way you'll get to hear it. Here is the call. Okay, can you hear this?
Yes.
Okay. Okay, so that's not something that you might think of as a bass note. You might not think that sounds like a very low-pitched call, but in cricket world and in bush cricket world, that is a really, really low-pitched call. Okay, and so with a call that's this low, it effectively is the lowest, one of the lowest frequency calls in the forest. forest. It's as if everybody's speaking like this in the same tone that I'm speaking now, and this animal is speaking at a very, very low pitch. Okay, so this is the equivalent of a very low bass voice, and with everything in ecology, having um, one kind of solution tends to produce more problems. And so one of the problems that I set out to investigate in the course of my postdoctoral work was if the males produce such a low call at 3.2 kilohertz, even though it comes from an entire family of high-frequency broadband callers, are the females adapted to hear such low sound? Okay, and that's not the paper that I'm going to be presenting today, but it leads up to my work. So I'll just show you kind of the summary of what we found in that paper for a couple of slides, and then we'll get into this paper. Okay? So the first thing that you need to know is that bush crickets and crickets have ears not on their head, but on their front legs. Okay, and you can see the white arrows pointing to the ears. And in the top right part of the screen, you can see a close-up of the ear. Okay, and the ear basically has a central stem and two kind of ear-like covers sticking out from the side. And there's not one eardrum, but two eardrums per ear. Okay, so we all have one eardrum, one tympanum per ear. But these animals have two eardrums per ear, and the ears are on the front leg. Okay, so what we found actually was that of the two eardrums, one eardrum, just listen to sound like everything else. We use vibrometry. One eardrum was tuned specifically to listening to the call of the male. So remember, I told you, if you look at the x-axis on the top graph, right, you find that the graph starts. Yeah.
We can't see the graphs. I don't think it's presenting correctly. We can just see the first slide.
You still can only see the first slide. Okay, then what I'm going to do is I'm going to. Can you see it now?
No.
No? Okay, I'm going to unshare and then I'm going to reshare. You see that?
We're still on the first slide.
That's odd because I can see I can see the second slide on what I'm showing you. Okay. Okay, I'm going to unscreen share. And then let's see. Okay, now you can see me. That means that you still see that first slide?
No, we can see you, but we can't see the. Can you hear us?
Okay, fine. So now let's see if you can see them. Can you see this now?
Hello. Yeah. Yeah, we can.
Can you? You see something else now?
Yeah, we see the slide.
Okay. Fine. So instead of doing okay, can you see it now?
Yes, yes.
Yes.
Now we can see the arrows.
Yeah, you see the ears on the front leg?
Yeah.
Okay. All right. So those are the ears on the front legs. And each ear has two, right? So you see a graph now?
Yes, yes.
Okay. So the graph on top, right? Hopefully you can see this high res enough, right? It shows you that when you play different sounds to the eardrum vibrates or sort of responds to the sound at different intensities, and this eardrum is acting like a low pass filter, which means that it's hearing sounds up to 3.5 kilohertz. Okay, and then at 3.5 kilohertz, it sharply stops being sensitive to higher frequency sounds. Okay, and the point at which it stops being sensitive is this blue line. If you see leaves in one color. All of the different symbols on the lower graph are different colors. Their the temporal pattern of their call is on the y-axis, and the frequency of their call is on the x-axis. And you can see that Onomachus, the red arrow points to this animal, Onomachus or whistler, right? It's one of the lowest, the most deep voices in the rainforest, and the ear is listening pretty much only to that call. It's blocking out all other calls from the rainforest. Right. So, one of the results that I was looking at, and then we found that, of directional responses. That is to say, we usually tell directionality using both our ears, right? We assess the direction of incoming sound by comparing the sound that arrives at one ear with the sound that arrives at the other ear. Like, where does the sound arrive first? That gives us a clue as to whether the source of the sound is on the same side as the ear oral, or between your time and intensity differences. And it turns out they can do this even with a single ear. So each eardrum on each ear by itself shows directional responses to another paper. Okay, so to follow up on, these are some of the things we found. We found these very nice co-evolved systems of the male call and the female response. The female ear hears only the male call and no other call, even a lineage where it usually listens only to high frequencies and not to lower frequencies. Call and the female eardrum have co-evolved, and that they show directional ability. And now what we want behavior to do to study, find she not. Right? So what happens?
Ma'am, can you repeat the last part? We didn't.
Okay. From here, right? From this slide?
No. Can you repeat the last part? We didn't catch that.
Okay. So I'm repeating from this slide, right? This behavior of Onomachus females. Do you want me to go to rewind? Okay. I'm just gonna go on. So we found that the ear was tuned to the male call, but we wanted to see how tightly the female's behavior was tuned to the ear call and compare female behavior with the ear's structural selectivity. Okay, so we wanted to find out what the direct frequency tuning of her behavioral responses to the male call. What is the frequency tuning? What is the directional ability that she shows behaviorally speaking? What is this the sound that the lowest sound that she's sensitive to? Okay, and these are the messages. We put an insect on a T-shaped stick, and we put a speaker on one side, which is playing the song, and an identical speaker on the other side of the T-shaped twig, which is not playing the call. Okay, so we just see whether the female walks down to the T junction and takes walks towards the speaker or not. We consider her to have performed. So typically, what happens with these animals is that the males call and the females walk towards the males. Okay, so we wanted to see. We considered her to have responded when she walked all the way to the T junction and walked all the way to the speaker playing the call. Okay, so we tried several different protocols for this, and this is what we found. I'm going to show you a movie now, so hopefully you're going to be able to see the movie. All right, play this. Now, I wanted to show you a better movie than this, but this movie is, I think, the best. Okay. I'm just going to open this up again, and then I think I'll have to share it separately with you. So I'm not going to show you more movies because it's very complicated. Okay, so can you see this this screen? Probably not. So I'm going to screen share. Just going to share the entire screen. Okay. Okay, can you see this? I'm going to start playing again from the beginning. I'm going to move this up. Can you see that shaking? Okay, so I'm turning into a. Okay, so did all of you see that video first of all? Yes, maybe. Okay, so all of you see, are you back to my slides? Can you see my slides now? Okay, so all right, so what we find is a very unusual response, which is that this is an example of a female who actually performs phonotaxis. She ends up walking towards the source of the call, okay, and you know, if you like, I can show it to you all the way till the end, and then you'll see her moving all the way to the source of the call, okay. But here the female sits and vibrates on top of this substrate, right, and she seems to vibrate kind of out of sync with the male call. And this is a video which I think I'm not going to be able to show you because of various system constraints, right? And so here's what happens. Typically, first I think I need for you guys to tell me, did all of you see the shaking? Yeah? Yes. Did you see the male call and the female kind of shake?
Yeah.
Okay, so the shaking typically happens for, you know, this animal just shook 2 or 3 times before walking to the T-junction, and then at the T-junction she turned, right? But most animals will first start this shaking, and it'll continue for up to almost 2 minutes, right? About 120 seconds on the time scale. And then she'll start doing phonotaxis, which means walking towards the source of the call, okay? And typically she goes from there to the T-junction. She tremulates some more at the T-junction, and then end of the towards the speaker. And very, very rarely she flies. Okay. Now it turns out tremulation, this kind of moving towards, you know, this kind of shake in response to the call, is a very, very specific response. So she only shows this kind of shudder, this kind of shake in response to the same species song. She doesn't do it when you play any other song to her, and she never does it in silence. Okay, phonotaxy, the animal just walks, right? Sometimes the animal will just walk towards, you know, one speaker or another, even in silence. So tremulation turns out to be much more specific than that, and it's also much more likely than phonotaxis. Okay, so if you look at the number of animals responding to with each different kind of response, all 19 out of 19 animals that we tested tremulated, that is, they shook in response to the call. But a significantly fewer numbers of animals walked towards the source of the call, and even fewer animals flew towards the source of the call. Okay, and as you can see, the number of animals performing phonotaxis is much lower, but also their accuracy is not very good. Okay, so these, you know, about 13 animals actually move towards the speaker, but only 9 animals move towards the correct speaker. Okay, and as you can see, even in response to silence or in heterospecific song, they sometimes move towards the speaker somewhat randomly. Okay, so so what we wondered is, given that tremulation is the immediate response, you can see it happens first, and it's a more specific response, and it's a less inaccurate response, right, than tremulation. We wondered if it had some kind of, I mean, obviously, if the animal's expending so much energy sitting around and dancing in response to the song instead of moving to a source of sound, maybe, you know, there is some kind of signal being sent out through this tremulation. And so we tried to see if it was a vibrational signal by, again, using a laser Doppler vibrometer, right? So we're playing the call to the animal, the male call to the animal, and we're also directing a laser onto the branch. Depending on the movement of this branch, so all of you know about the Doppler effect. So when you send, when a signal is either reflected from or produced from a surface that's moving towards you or away from you, it changes the frequency of the sound that reaches, or you know, in some sense, it will pick up that perpendicular motion through shifts in the frequency of the laser that the reflector. What we found when we did this, you can see, I mean, I think I could have shown you other videos which were, you know, bulkier than this one, which I couldn't transfer to this computer, which show this very nicely. But you see that there's a very nice alternation of male call, female tremulation, male call, tremulation, call, tremulation. Now, as you can see, the tremulation varies in its strength. But what with the tremulation, and you can see this close up as well, male call, female tremulation. This delay between the starting of the male call and the starting of the tremulation, that appears to be an extremely tight, very, 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 very conserved aspect of the female response. You can see that the female call, as I've told you before, is a pure tone call, peaking at about 3.2 kilohertz. Right, and this tremulation signal also has a certain, I mean, it's not as clean, but it also has a characteristic frequency which is about somewhere between 40 and 50 hertz. Okay. All right. So this kind of back and forth signaling is basically a duet, right? And it's actually a fairly nice and novel multimodal duet. We know a fair amount actually about acoustic duets between insects and frogs and birds, right? We know about vibratory duets between spiders and lacewings and plant hoppers and stoneflies. We know that there are generally multimodal courtship signals, but we actually don't know of any other multimodal duetting where one animal sends a song in one modality, that is sound, and the other animal responds to each call with a specific temporal structure, which is in a completely different modality, in this case, vibration. Okay, and as you can see, for each animal, right, so if you consider this portion, from one male chirp to the next male chirp, or from here to here, as one side chirp, then for the first sort of good, over sort of less than a quarter of the cycle, the male chirp, continues, and then each of these arrows represents the phase of the timing of the female's response to the male call. Okay, and this is a phase vector that represents the sum of all of the phase vectors of the different phases of the female's response. So the fact that each phase vector is very long suggests that all of the vectors are roughly aligned. Okay? So it looks like there's a difference between different individual females about how delayed their shaking is relative to the male call. Can all of you still hear? Yeah? Okay, everyone's looking attentive, so I'm just going to keep talking. Okay. So each individual female has a really tightly tuned, latency of her response to the male call, although there is some variation between females. Okay. So what is the point of all of this love song? Like, he's calling, she's calling. Eventually, someone has to find someone else and mate, right? And we know that females show a very large spread in terms of how long they tremulate before moving towards the male. Okay. So the question is, first of all, can the male localize the female? So we did this in a fairly simple way. We put the male, on one end of not the T-shaped branch now, but we needed a nice branch which would kind of, somehow we needed a female on the branch. But if we just put the female on the branch directly, we ran the risk of the female, not just sending a vibrational signal, but also sending olfactory signals when she shakes, or doing very thought that it would be a good idea to put the female in a box, right? So one box contains the female, and we needed a branch that would nicely curve and kind of couple to the box. And then the other extreme end would be placed in an empty box. Okay, and the female has to make two choices. She needs to go to this Y shape and make one correct choice. The first choice needs to be correct, and at the second branch also she needs to make the correct choice. And since there are only two branches over here, we regarded her as having made a correct choice if she made two first correct choices. So the male, the male was able to move towards the female who's, and significantly more so than when the box was like she was there in the box, but the box was not connected to the branch, or a control where there was silence. Okay. We also we thought, okay, it's good that the male is able to move towards the vibrating female, but we wanted to be sure that maybe the box was the box had traces of sexy female smells on it. So we really wanted to make sure that the male was moving towards the vibratory signal. And so we then we took pretty much the same setup, except we replaced the box containing the female with a piezo, which is actually, we really just use a large bass speaker. Okay, and we just ran the signal, this 40 to 50 this 40 to 50 hertz vibrational signal to the bass speaker. Okay, and then we played the vibrational signal to the male. Now here's where we were surprised. It turns out that when you play just the vibrational signal to the male, he simply does not move. It's not like he moves around a little bit. He just doesn't budge. He acts as if he didn't hear the vibrational signal at all. So we were then a little worried, and we thought, well, he's clearly moving towards the female when she's vibrating in the box, and we're pretty sure that it's not smell that's the attraction. But just to be on the safe side, we tried a trial. Can you hear?
Yeah, no. I was asking, the vibration you provided, was it in phase? Was it like in the same phase as the female provides?
Yeah. So, phase is all relative, right? The exact same structure, and frequency based of the vibrational, and then those were played back in the exact natural phase relationship they have to one another. So then we have the male call from the speaker and the female vibration being artificially played through the bass speaker, both to the male. And what we found is that when you play the vibration plus the sound, 100% of the time, the male moves towards the source of the vibration. And I can possibly even show you a video. Um, I'm going to try. Okay. Okay, so here's the male, and you will only hear the male's call being played back to this male, and the vibration is played back out of phase with the male call. Okay, so you can see that the male kind of samples a couple of branches, tries to figure out where the vibration might be coming from. I think he decides it's coming from here. Stop really moving very much, and moving, he needs to make a second choice of the second branch. All of those sounds are the sounds of me moving the video camera to track the male. Okay, again, no sound, no vibration, he's not really moving. And then we'll start up the sound and the vibration once again, and you'll see him. There we go. Okay, and he looks pretty motivated now. Bass speaker. Okay, you had. Okay, right. So we basically therefore find that he needs both components of the duet to move towards this, not move to the vibration only, and this is an extremely significant result. Okay, and certainly, we also tried several other controls to make sure, when you see this video, especially when you can't see the vibration but you can hear the sound, you would think that maybe the male is responding to the male call and not to the female vibration. Well, when we play sound only, the male doesn't move. When we play the sound and the vibration, but that speaker, that bass speaker which you saw is disconnected from the branch, right? Then again, he doesn't move. And of course, in a control where you play neither sound nor vibration, he doesn't move. Okay, so this is a very significant result. It signifies that he will move towards a source of vibration in what we're calling vibrotaxis, as long as the vibration reaches him in a specific phase relationship to the male call. Okay, so he'll move towards a source of vibration regardless of whether it's a tremulating female or a vibrational signal. So they are responding not to the vibration but to the duet, right? And both components of this acoustic vibratory duet are therefore required to for him to functionally locate the source of the vibration. All right. So this therefore means that there are two loops of communication occurring between these animals, right? The male calls, right? This can lead either to female tremulation or to female phonotaxis. If the female is performing phonotaxis, then she actually just moves towards the male. Meanwhile, the tremulation, sort of provides a feedback to the male, right? A vibrational signal, and then the male can perform vibrotaxis towards the female. Okay, so there are essentially two loops of communication happening between the males and females in this setup. So this leads us to obviously ask some questions. One question is, why tremulate, right? Why not just, why does the female just not move towards the male? Well, with a lot of why questions in evolutionary biology, we can't answer them definitively, but we can look at correlative ecological factors that might have produced this system or that seem to make this system advantageous. One is that, this is another reason that this entire duet, this system that we discovered is extremely rare, is because this kind of tremulation or shaking is usually not seen among females also occasionally of South and Central America, where it's been shown that bats are able to locate calling males using their calling song. So you can play the calling song to a bat, and the bat will come and try and prey upon the speaker from which the calling song is played. Okay, among in our part of the world, for example, in India and in Asia and in Africa, crickets do not seem to, like the sounds of crickets don't seem to attract bats with such a high probability. Okay, and therefore, seem to not be so specialized to listen to sounds. And we know that bats can either echolocate moving insects or use the sounds of flapping wings to find the moving insects. 100% of flying females are. Now, data suggests that in droppings, in the droppings of Kudremukh bats, because kind of either spit them out. I mean, they basically tear off the leaves before eating the crickets. The use of tremulation as a preferred channel of private communication is used by male bush crickets in the neotropics, but in the paleotropics, where we find that females who are actually affected by predator pressure when they are moving, it looks like the predator pressure here has shifted away from calling males onto moving females. And communication from the female to male, essentially signaling that she is under risk and cannot move, and he's under less risk as a caller and should move towards her. Burden and onto now, tremulation also walking and noise from the vibrational channel also seems to be less than the acoustic noise in competition that one faces in the cocktail party of the rainforest. I've also done a bunch of studies looking at the choice between tremulation and phonotaxis. That's been written up and it's for it to get published. But basically tremulation always seems to be the first response. You want to look at whether either the number of animals tremulating or the latency with which tremulation and phonotaxis are switched between correlate with various parameters of real songs, such as the loudness of the song or the frequency of the song. And one of the other things that we're looking at now is what is the moving mechanism producing frequency to the male call. And off with tremulatory response, so working on than phonotaxis. We want to look at what kinds of signal propagation distances characterize each arm of these communicational loops. What is the directional ability of the male while performing vibratotaxis performing phonotaxis and so on. And we have some other systems that we're also working on just among crickets and bush cricket. Ability to hear itself in the crowd. Predator-prey interactions and differential tuning of neurons to detect the same species song versus the bats. And PhD now also working on another bush cricket called Nicopoda, which produces several different song types. And we're looking at how those song types are generated and how receivers distinguish between these different song types. Right? So these are my acknowledgements. And can you hear me?
Not the last part, ma'am.
Okay, I just said I'm done. I'm done, and I can take
Hello.
questions now.
Hello.
Hello?
Yeah. The end got a bit patchy, so we can't really hear.
Okay. Talking about future projections and collaborations.
Hi, can you hear us?
Yeah, I can hear.
I guess it's done. It's, I suppose the other fantastic. Let's thank the speakers are
Yes.
that's how it is. Thank you, ma'am, for agreeing to join us for this webinar session. It's as you can see, it's quite new and the net is a bit patchy, so we had a bit of difficulties here and there.
I think it's great. It's really important to try and arrange talks remotely.
But I guess most of the message did get through.
Excellent. And I will hopefully be there sometime over next year to present this and other works.
Then we look forward to hearing you with better quality then.
Lovely, lovely. Does anyone have any questions?
I'm sorry? I'm sorry?
Does anyone have any questions?
Oh, questions? Any questions? Actually, you can come up here and probably.
I'm glad that people are here.
What I'll do is I'll give you the. Yeah, she can see you.
Yes, I can see you.
Yeah, speak up, speak up.
Ma'am, the question was that, ma'am, you were saying that for a perfect response from the female, both phonotaxis and vibrotaxis should happen equally because both should happen consecutively. But ma'am, for the phonotaxis, the disadvantage was that bats may be attracted to that, and they may feed on the male insects. So why does both of them then happen? Then shouldn't only vibrotaxis should take place, because that is more beneficial. Why phonotaxis then?
Right, that's a good question. Yeah. Well, two answers to that. One very common thing is, suppose the male is calling from another tree, right? And the female is vibrating from this tree. So can you hear me?
Yes.
Well,
yes.
So if the male is calling from a different tree and the female is on a different tree, it's never going to, right? So the vibration is something that works when both are on the same tree.
Vibration works when both are on the same tree, but supposing the other one is on the other.
Right. So that's one answer. And the second question is that if they are on the same tree, so one of the, the next piece of work designed around this, which was that we wanted to see if the male doesn't, if it sounds like the male is really close, if the male's call is really loud, does the female preferentially push the male is nearby? And it turns out that the At all loudnesses of the call, the female tremulates first. Okay, I'm back.
Yeah.
Right. So actually, the female always tremulates first, and eventually the male. So it looks like there are reasons for both communication systems to still be in the loop, but the other thing is that this animal may still be a work in progress evolutionarily, and we're also interested, of course, in tracking the animal over time and seeing what changes. Did that answer your question, and could you hear everything?
Okay, ma'am. Ma'am, in the case of the first question where she can you hear me, ma'am? Okay.
Go on, go on.
Ma'am, in the first part which you said that if both of them are not present on the same surface when vibrotaxis cannot work, in that case, is phonotaxis enough for one of them to approach the other? Because there will be no vibrotaxis.
Yes, absolutely. To the male eventually, but as I said, it's a delayed response, it's a non-specific response, and she also makes errors. So yeah, I mean, one would think that if they're on different trees and vibrotaxis doesn't work, that the female can eventually move. And note that we have also looked at phonotaxis by walking versus flying. And so we find that only 4 out of 19 males were willing to fly at all, right? So even if the females are moving on the same tree and to fly, so it looks like in this system, females are really trying to conserve their movement as much as possible between trees. But certainly that's the only thing that will work if the male and female are on different trees. And I also predict that therefore, the state of communication of these animals would vary in ecological settings where they are high in concentration versus low concentration. That's another ecological study that we're doing, because we actually find these katydids in several parts of we looked in a village actually, which is near Kudremukh forest, and that's for a very practical reason. Because, in the rainforest, the trees are too large. So, but if one were to look at plantations where these jackfruit trees were even more far away from one another, or smaller jackfruit trees, we would probably find different patterns of choice between. It's possible that we could preferences of tremulation versus phonotaxis.
Thank you.
Next question.
Any questions?
Hi, ma'am. If there are like multiple females or males, then how do they resolve it? Like there will be multiple females in calls and vibrotaxis. So
Right. That's a good question also, and we don't really know how mate choice operates, either for females when there are multiple males around, or for males when there are multiple females. This is something that we are going to have to look at. I mean, the good thing about this study is that it's in a completely new animal, and so there are tons of questions that one can ask and should ask, and all that we need is lots of students who are interested in working on it.
okay.
Ma'am?
Yeah.
No, there is obviously, when the females tremulate, there's obviously a delay from the male call, right?
Yes.
So have you played around with how long you can delay this, as in when the male responds?
Yeah, these are all very good questions. This is what we're hopefully going to be working on this season.
Okay.
But she decided, or we decided, I guess, that she should do field work and try and figure out how the males are. We can figure out the ecological significance in what it means to be calling, how many males might be calling per tree, let's say. And the other so what we will do then is take males and see whether messing with the timing, how much you can mess with the timing of the female stimulation signal relative to the male stimulation calling signal performed by the
And
male. And
this,
actually, we also will do the that, which is mess with the male call structure and see how much we can change the temporal pattern of the male call before the female responds with stimulation. We've already done that with respect to frequency, but we'll do it with respect to temporal pattern.
thank you.
Yeah.
Any more? I guess there are no more questions.
Fantastic. Very good question.
Anyway, thank you, ma'am, for joining us tonight.
All right, my pleasure. Take care. And hopefully I'll see you all in person.
Yeah, we are.
Okay. Bye bye.
Okay. Bye.